For the third and last part of our discussion for this module, we're going to talk about the invisible enemy that's lurking around us at this very moment. During the time of pandemic, it is very vital for us to know something about the presence of these viruses. On our discussion for this module, we are not going to discuss yet here the epidemiology and the presence of the possible vaccines. We're just going to establish first the different concepts and facts, and we're going to adapt the discussions that we had during Module 1 and Module 2 and the earlier parts of our discussion in this video presentation. Viruses are very small infectious particles consisting of nucleic acid enclosed in a protein coat and, in some cases, a membranous envelope. As you can observe here, E. coli bacteria and the phages or the bacteriophage. Whenever we are talking about viruses, they're actually considered as obligate intracellular parasites. Viruses are different types of parasites that is coming from the exterior of our body, and they need a host. They need a certain type of organism in which they are going to be attracted with, and at the same time will be responsible for the creation of more types of viruses. In this case, whenever we are talking about viruses, without the presence of the cells that we had discussed earlier, both eukaryotic and prokaryotic cells, viruses will not be present. There is actually a debate when it comes to the evolution of the different viruses. Did the different types of viruses, even though they are non-living things, evolve because of the presence of cells? Or does cells evolve because of the presence of the different viruses? Different scientists, epidemiologists, microbiologists, and even virologists, virology, which is the study of viruses, are considering here the possibility that cells and viruses are actually coexisting, even in the earlier formations of the different type forms on Earth. But at this very moment, we're just going to establish once again different facts. Let's start with the different structures that is present among viruses. We're going to consider here the different parts that is present on every single one of them. A typical virus is actually possessing here the genetic information. It could either be in the form of an RNA or the presence of a DNA. Then at the same time, there is the presence of the capsid or the protective layer that is present on all viruses. This capsid is actually made out of proteins and other complex materials that will simply protect the interior of the virus known as the genetic information. And some of these viruses are actually possessing here what we call as an envelope. And this envelope is possessing different types of glycoproteins that will now interact with the different cells on our body. If you can still recall on our discussion when it comes to the plasma membrane, the plasma membrane is not only possessing phospholipids, but also the presence of other types of carbohydrates and even proteins, and even the presence of glycoproteins, just like what is present among different types of viruses. When it comes to their shape, we have here the presence of a helical-shaped capsid. Then the next one, we have here the presence of an icosahedral shape. Then the next one, we have here the presence of a helical capsid which is circular. And lastly, we have here the presence of the icosahedral head and the presence of the helical tail. And the examples of these viruses are the following: tobacco mosaic virus. Then the next one, the presence of adenovirus. Then the third one, influenza virus, and even coronavirus is included for the third classification. And lastly, the presence of the bacteriophage, the one that is responsible for infecting bacteria. In the case of viral replication, viruses are obligate intracellular parasites that can simply replicate within a host cell. Without the presence of prokaryotic or eukaryotic cells, viruses cannot replicate themselves. When it comes to the concept of infectious diseases, we are using here actually the term known as the R0, or what is the probability or the possibility that a person can infect other person. In the case of viruses, they're actually using here the host range, or it is now the capacity of the cell to infect other types of cells. The very first thing that will simply happen here is the presence of the entry and uncoating. If this is the presence of a host cell, the virus will simply enter that particular cell by either injecting its DNA or the incorporation of the virus itself. Okay, once again, it is case-to-case basis. Just like the different organisms, viruses adapted to the different change that is happening in your environment. The second thing that will happen after the entry and uncoating, it will be replication. The genetic information present now on the viruses will now be replicated inside the cell. Then, after the replication of the DNA or of the genetic makeup, it will now proceed with the transcription and the manufacture of capsid proteins. Remember the concept of central dogma of molecular genetics. It is also happening on viruses. They will now transcribe different types of materials and will now create capsid proteins. And these capsid proteins will be very vital when it comes to the formation and self-assembly of new virus particles and their exit on cells. Cells can actually die in the process of viral replication. And when it comes to this particular life cycle, it is present on bacteriophage, which is known as the lytic and the lysogenic cycles. For summary, we have here the presence of the lytic stage. In the case of the lytic, from the name itself, the cell will die or the cell will lyse. From the name itself, lyse, which simply means the destruction of a cell. So the very first thing that will happen during the lytic cycle is the attachment of the bacteriophage, then the entry of the DNA on the host bacterium. Then, when this is happening, the DNA will now be destroyed. And when the DNA of the bacteria is already destroyed, it will now incorporate the DNA that is coming from the bacteriophage. Then, the third part is now the synthesis of viral genomes and proteins. Because the DNA is already injected, the cell itself will now undergo the process of the creation of the necessary proteins that will simply create now the new materials necessary for the formation of new viruses. Then, assembly will happen with the presence of the head, the tail, and the tail fibers, and lastly, the death of the cell by means of the release of these newly created phages or bacteriophages. That's the thing about lytic cycle. Lytic cycle is happening only once, then the cell will die. That's the thing about the lytic cycle. Okay, attachment, entry, synthesis, assembly, then release. Unlike in the case of the lysogenic process, the cells of that bacteria will not die instantly. But rather, if a cell of a bacteria will be injected with the DNA, it will now undergo the reproduction process, binary fission might happen, and those new types of bacteria will contain the same DNA that is present on the bacteriophage. Then, when the trigger mechanism starts, the lytic cycle will be induced. And as a summary of the lytic and the lysogenic process, lytic cycle is happening on virulent or temperate phages. What are the different types of virulent phages? These are the different types of viruses that kills a bacteria instantly. That's the term about virulent. And it can spread rapidly. Then, destruction of host DNA. Then, production of new phages and lysis of host cell causes release of progeny phages. Or whenever we're talking about progeny phages, these are the offspring phages. Now, in the case of the lysogenic cycle, they are temperate phage only, or these are the different types of phages which are not that destructive, but still they have the capacity to harm that bacteria and definitely kill a bacteria. Then the genome integrates into bacterial chromosome as prophage, which is replicated and passed on to daughter cells during the binary fission and can be induced to leave the chromosome and initiate a lytic cycle. Or technically, it can now initiate the death of that bacterium. So that's the difference between lytic and lysogenic cycle. And once again, this is happening only among bacteriophages. Now, we have the different infectious agents, viroids and prions. Viroids are small circular RNA molecules that infect plants and disrupt their growth. Unlike viruses, which are somewhat proactive whenever there's the process of replication and infection, in the case of viroids, they're actually somewhat passive. They are just being passed on from one part to the next by different infectious agents. It could be by means of the presence of the different vectors or the different organisms that carry these different types of agents. Now, we have here an example known as the potato spindle tuber viroid. These potatoes are infected by this viroid. They are not infected by a specific virus, but only an agent. Only circular RNA molecules that not only potatoes are being affected by the potato spindle tuber viroid. It is actually attacking the family known as the Solanaceae or the Solanum family. Not only potatoes are under the Solanum family, but also tomatoes. Tomatoes can also be infected by this viroid. Other than the infectious agents known as viroids for plants, there are also different infectious agents that are now present among humans, and they are known as prions. Prions are slow-acting, virtually indestructible infectious proteins that cause brain diseases in mammals. And as you can observe here, this actually happened on the late 1990s and even on early 2000s in European countries, specifically on United Kingdom, known as the mad cow disease. It is caused by prions that targets the soft tissue that is present on our nerves. The reason why they are considered as mad cow, because they are possessing here symptoms of restlessness, then fear of other organisms, fear to eat different food products, and even fear of water. Or technically, from the name itself, mad cow, they are like zombie-like symptoms. Okay, in the case of the mad cow disease, it is also known as the bovine spongiform encephalopathy, or it is the disease that targets the brain of those cows. The reason why they are possessing different types of symptoms. And scientists are actually arguing that the so-called zombie apocalypse that is mentioned on different sci-fi films, sci-fi TV series, can actually happen not because of viruses, but of simpler infectious agents known as prions. Remember prions, just like what is happening in this particular cow, there is the presence of this brain disease that targets the nerves of our brain. Because our brain is actually possessing here different types of proteins, and the prions are attacking those types of proteins. And even humans can have the same symptoms, not only in this particular prions, but also even on some viruses, just like the presence of the rabies virus. But rabies is a virus and not a prion. Then, if we're going to consider this particular type of zombie apocalypse, I actually attached a very interesting video presentation regarding the possibility of those things, because we never know. Maybe sooner or later, the science fiction that we know will be the reality that the future will simply face.